So girls, today we will start with a new topic, okay, on enzymes, that is enzyme kinetics. And you will see in your syllabus also there this topic, enzyme kinetics for catalysis. So first, let us try to understand what do we mean by kinetics, by enzyme kinetics. Now, as you can see here that the enzyme kinetics, it refers to the study of the rate of enzyme catalyzed reactions. Okay, it is the study of the rate of enzyme catalyzed reactions, meaning those reactions which are catalyzed by the enzymes. What is the rate? Okay, how it is, how it proceeds. What is the, suppose whether the that particular reaction, whether it is slow or it is fast, what is the rate, okay? And also not only the rate of the reaction but also the effect of various parameters on it. Now I had already sent you some videos on those parameters which affect the activity of the enzyme. For example, pH, then we have temperature, we have inhibitors, and so many others. I have sent in that video if you have watched. So enzyme kinetics is not actually only the study of the rate of the reaction but also the effect of all those parameters on the enzyme. Okay, so now the rate of such reactions is measured in terms of number of product molecules formed. You see here that the rate is measured in terms of the product molecules formed, or we can see the substrate molecules which have been consumed per unit time. Here you see per second. So just remember that how that particular rate, how it is measured, the rate of any reaction which is catalyzed by the enzyme is measured through the number of products formed, because we know that when the substrate molecules they will combine, okay, they will give rise to the formation of the products. So it depends on the number of product molecules formed, or how many number of substrates have been consumed during that particular chemical reaction. So that is the rate. Okay, it is measured in terms of the product or in terms of the substrate. Now to understand that better, we have to see we have to plot the graph for this, for the enzyme catalyzed reaction, and we'll try to understand that particular uh, graph. Okay, so now for example girls, just listen this carefully. Example, now just imagine I took a beaker. Okay, I take a beaker, and inside that beaker, suppose that is just for an example, okay, just for you to understand. Suppose you have 100 molecules of enzymes in that beaker, and outside the beaker, suppose I have many substrate molecules. Okay, inside the beaker I have 100 molecules of enzyme. Just try to understand this. Now what happened when I give the, suppose I insert in that beaker, suppose I keep 10 molecules of substrate. Alright, now those 10 molecules of substrates, when you keep them inside that beaker where 100 molecules of enzymes are present, you know that the substrate will go and bind to the enzyme, right, to form the enzyme substrate complex, the ES complex. So since I have 10 molecules of substrates, that means those 10 molecules will go and bind to the 10 molecules of enzymes. So 90 enzymes will be free. I hope you understand this, right? So that is why this first line here is said that when the substrate concentration is very small, only few enzymes will form the enzyme substrate complex, while the most of the enzyme molecules will remain free. Okay, and the number of products molecules form also will be few, because only 10 enzymes has bind to the 10 substrates. They will catalyze the reaction and those substrates will form the product. So at this stage only few product is formed. Okay, so this stage where only few enzymes are bind with few substrates to give rise to few molecules of product, at this stage the rate of enzyme catalysis, or we call it the initial velocity, okay, the speed of the reaction is given by V naught. Okay, the rate or the speed of a reaction is given by V naught. All right, so to understand that, just take a look at this graph here. Now you see here the V naught, that means the initial velocity. Okay, when you add small amount of substrate in that beaker, only few substrate will go and bind to few enzyme, while the rest of the enzymes will be free. So when you plot the graph, it will be this one. Okay, so this is the initial velocity. It will start to increase slowly. Okay, it will start to increase slowly. As long as you are added more and more substrate, those substrates will go and bind to more and more of the enzymes. But remember, as an example, I have given you that in that beaker I have only 100 molecules of enzymes. So as long as some enzymes are free, so if you add more and more substrates, the rate of the reaction also will start to increase linearly. But the velocity here is referred to as the initial velocity and is given by V naught. Okay, so at relatively lower substrate concentration, V naught increases almost linearly, but with an increase in substrate concentration. Now what happens if I add more and more and more concentration? So this V naught will increase smaller by smaller, and finally you can see a point is reached where it beyond which it increase no further. Okay, till here. Till here it will not increase anymore and it will become a straight line. Even if you add more substrate concentration, it will become straight line. Why? You must be thinking why. Now remember, in a beaker I have only 100 molecules of enzymes, right? So if I add more and more substrates, suppose all those all those enzymes, they have been occupied by the substrate molecules. So there is no point if I add more again, because if you add more substrate, no enzyme is free at that time. Okay, at this point there is no enzyme which is free, because all the enzymes are occupied by the substrates, all the because you increase more and more, okay, the substrate concentration. When you increase the substrate concentration, at first the graph also, this V naught, it will increase linearly, but a time will come when no enzyme is free to bind with the substrate. So even if I added more substrate to that beaker, when you plot the graph, it will increase no further, because those substrates which you add later, which you add more, those will have to wait for some enzyme to freeze, so that it will go and bind to that enzyme, so that a chemical reaction will occur. So this maximum, that means it has reached a maximum velocity at this point. So that maximum velocity is represented by V max. Okay, so this point is termed as Vmax, that is a point at which the velocity now it has become maximum. And this kinetic behavior of an enzyme catalyzed reaction, it led Victor Henry, okay, this scientist Victor Henry, in the year 1903 to propose that the formation of an enzyme substrate complex, that is the ES complex, is the necessary step in any enzyme catalyzed reaction. Because like I had mentioned to you, that even if you add more and more substrate concentration to that beaker, a time will come where the reaction will increase no further. That means it has reached its maximum velocity. Why? Because at that time no enzyme is free anymore because there are only 100 molecules of enzymes in that beaker, so no enzymes is free because all those has bind to the substrate. So at that point there is no ES formation complex even if I add more substrate to that beaker. So this ES complex formation is the necessary step in any enzyme catalyzed reaction. Just remember that. So this proposed idea of Victor Henry was done being expanded by Michaelis and Menten in the year 1913 in their model. So now we are going to study the Michaelis-Menten equation as well as its derivation. So okay, from this graph, I hope you understand when the substrate concentration at initially when you add few substrate concentration, then this initial velocity it will start to increase slowly. Sorry, it will start to increase slowly. All right. So slowly by slowly it will start to increase and finally it will reach a point where no enzyme substrate complex will form because all the enzymes have been occupied. So at that velocity it has reached its maximum velocity referred to as the Vmax, and any further increase in the substrate concentration, if you add more and more substrate at this point at this Vmax, it will not change the rate because the enzyme molecule is not free to form the ES complex. Okay, so this is the saturation stage and you can see that the graph it forms a plateau, and when the substrate is in excess the product concentration also it will gradually increase, okay, because so many substrates are there. So even the product concentration it will start to increase gradually and then it will reach its maximum. So at this stage the rate of forward reaction, that is substrate to product formation, is equal to the rate of backward reaction, that is product to substrate. Okay, substrate to product and products to substrate. So this forward and backward reaction they are equal and this stage is referred to as the equilibrium stage. You can see here this stage is referred to as the equilibrium state. So we are going to study the Michaelis-Menten equation to see the effect of this ES formation complex on the rate of the reaction.